Hi, folks. Today we're going to talk about patterns of diversification through time among the major clades of the neotropical freshwater fishes. And this talk begins with a big question, and that is, why are some clade species rich, but others are not? Are these clades characterized by faster rates of speciation, capable of generating more species in less time than others? Are there common ecological and intrinsic factors in these clades that make them more predisposed to high speciation rates? Things like small body size, ancient origin, Wrightian population demographics, and key innovations that make them capable of speciating rapidly and generating high species richness. In other words, does ecology, things like habitat and diet niches, explain species richness in some clades? Or on the other hand, is biogeography a better explanation for high species richness? The classical model for this is that genetic drift occurs when populations of a species exist in allopatry, and then when the barriers break down between those populations, reinforcement takes place in sympatry, leading to speciation. And this is obviously accelerated by landscape evolution, things like river capture and fluctuations in sea level. And so to answer this question, we're going to look at 13 major clades of neotropical freshwater fishes. And I want you to notice the range of species richnesses present in these clades and the range in their crown ages. And also combined, all 13 clades here represent 92% of the known species of neotropical freshwater fishes. So the first result I want us to look at here is the per lineage speciation rates for each species in the tree. And this is the result from a model called clades. And what I want you to notice is that many of those clades we're interested in are characterized by high rates of speciation, shown here in warmer colors. But at the same time, there are a lot of low speciation rate clades present here as well. And so over the top of this, I've overlaid the results from a model called TESS, which models the total diversity of all of the species in the tree through time. And what's immediately striking here is that at around 20 to 30 million years ago, there's a large increase in the diversification rate. And this also coincides with the crown ages of a lot of the most species-rich clades, which makes sense. And so for a bit of biogeographic context, I've shown you some maps of South America at that time. And there are two major biogeographic events that I want you to be aware of here. The first is that between about 50 and 30 million years ago, this continent is characterized by the Lago Pebas mega wetland system. And then between about 30 and 10 million years ago, we see the Amazon River capture event, which led to the current modern drainage of the Amazon River. And you'll see a lot more of this in the talk following mine that focuses on biodiversity or on biogeographic patterns. But for now, just bear those two major events in mind. And finally, when we look at this result, I want you to keep a couple key caveats in the back of your head. The first is that up until about 100 million years ago, there actually aren't very many extant lineages of neotropical freshwater fishes. And that makes estimates of their diversification rate a bit weaker than they are towards the present. Next, that big spike we see at about 20 to 30 million years ago, the spike in diversification rate, that is going to be influenced by the data richness of the alignment that was used to build that tree. If there simply aren't as many bases in the genomes and the genetic data that's being used to build this tree, we're going to see a much higher inferred rate. It's going to bias it upward. And finally, the decline we see towards the present, which I've highlighted in a red triangle here, is mostly due to the taxonomic deficit. We've only really described about 2/3 of the neotropical freshwater fishes. So those are all going to fill in that triangle. And so this should be seen as a call to get out there and describe more species, right? So if we can bear those caveats in mind, I think we can ask the question, to what extent are these major clades characterized by the ecological factors that we tend to associate with high species richness? Things like body size, ancient origin, Wrightian population demographics, and key adaptations. And so let's take a look at a couple major clades here. The first is the Loricarioidea, which are characterized by a unique feeding adaptation. Within this clade, you see that hypostomus is actually speciating much, much more rapidly and has higher species richness than many of the other clades within this tree. And so of those 2 or 3 major radiations within this clade, we see that they actually seem to correspond roughly to the Megapebas wetland system and the river capture event present in the Amazon. Similarly, in the gymnotiformes, which are characterized by a unique sensory adaptation, we see that some clades, like the Stenarchorhynchus plus the Navahini and the Gymnotus carapo species group, are characterized by rapid rates of speciation and higher species richnesses, whereas there are other clades that are actually very slow and relatively species poor. And these are also characterized by the same biogeographic patterns that we saw before. And finally, the Curametoidea, which we're going to talk a lot more about in the next presentation, are characterized by a rapid radiation of Ceras salmus. This is relatively young, coinciding more with the Amazon River capture event, but overall similar patterns in the distribution of speciation rates among species in this clade are present. And so now let's take a look at some summary statistics comprising all 13 clades present. And the first major pattern that I'm curious here about is that species richness is not well correlated with the crown age of these clades. So while some of these clades are relatively clock-like in their speciation rate, many, many outliers exist. And so this begs the question, what factors are responsible for these clades that seem to be greater or lower richness than their age would lend us to believe? And we can break this pattern down by habitat type as well. In stream habitats, we actually see that the relationship between crown age and species richness is pretty good. But in river habitats, the trend is even reversed, likely due to a low variation in crown ages among these habitats, and probably a low species richness variation as well. And so what this tells us is that crown age is probably necessary, but certainly not sufficient for high species richness. In fact, many ancient clades are still relatively species poor. I'm sure you're aware of the lungfish and many other very old but species poor lineages. Next, I want to take a look at the inferred speciation rate for each of these models. This will be the average rate coming from clades and tests for each of our 13 major groups, and see how that relates to the number of species generated per million years. And what we see is that many of these clades are actually higher or lower in speciation rate than we would expect based on their richness per time. In other words, time alone still doesn't predict speciation rate or species richness particularly well. That trend is a bit stronger in rivers, where we see a higher speciation rate correlating with a high number of species per million years. But in streams, there are actually a lot of clades with low average speciation rates that produce a higher number of species per time. And this is because the average rate inferred from clades or tests is going to hide some of those extremely fast speciators and lead to an overall lower speciation rate than you see in the absolutely most species rich, highest speciating clades within a whole clade. And next, I want to take a look at that key period of time from about 20 to 30 million years ago, where we saw a lot of our clades originating on the whole tree. And what the major takeaway here is that there's actually a lot of heterogeneity in the pattern of speciation present at this time. What these test results are showing us is that some clades like the Gymnotiformes or the Lorocaryoidea are speciating rapidly at this time, as you might expect. But there are other clades like the Cyclinae that are experiencing the opposite trend. And in fact, many of them are actually relatively constant in time. So overall, there's a great heterogeneity of rates on a clade by clade basis within the whole tree. And so this takes us to some major conclusions. The first being that species richness is not strongly correlated with age for most of our clades. Within the clades, species level speciation rates are gamma distributed, not uniform. Some subclades are simply speciating much faster than others. Stream habitats tend to produce more species-rich clades than river habitats, and that the clade-wise average inferred speciation rate does not predict richness particularly well because of rate heterogeneity among subclades. We also note that several species-rich clades emerged or at least accelerated in their diversification around 30 to 20 million years ago. And this is coincident with the period in time characterized by the Lago Pebas mega wetland system and the Amazon River capture event. As we predicted, many species-rich clades are in fact small-bodied, and some have unique functional adaptations that we might associate with high richness. But within these clades, there remains an uneven distribution of speciation rates. So while large clades may have high average speciation rates and high richnesses, many subclades within those large clades remain slow and species poor. In other words, some subclades are speciating, but some are not. And so this pattern of speciation richness through time is described as what we call a series of fortunate events. And these include clades with the appropriate traits that predispose them to speciating, and also landscapes with just the right amount of geographic rearrangement through time. In other words, species richness cannot be explained just by ecology alone, things like habitat and diet niches. Functional redundancy exists within ecologically important traits. In fact, all of the traits that we looked at apply equally to all of the members of those clades, and yet some are speciating rapidly and some are speciating poorly. In fact, most closely related species tend to differ in adaptively neutral traits, like things that have to do with sexual recognition. Functional differences do exist within some clades, but it is often recovered that the rate of speciation is in fact faster than the rate of niche evolution. So we can't fully explain the differences in species richness using just ecological factors. And so this leads us to a model of species richness that includes both external factors, things like the biogeography of the continent at the time, as well as internal factors to the species as well, things like their unique adaptations. In other words, this is an integrational model. We have to include both externalism and internalism to give us integrationism. And the big takeaway I have from all of this as a taxonomist is that the taxonomy is not complete. The more species we describe, the more genetic data we collect, and the better trees we build, the better we'll be able to answer these big questions. And so to me, this is a call to get out there and describe more species. And so with that, I'd like to thank James Albert for inviting me into this project and helping organize this conference. I'd like to thank my co-authors on the HENF project who have collaborated with me and helped me organize some of this material, some of my mentors at my current institution and my colleagues, and of course, the organizers of this conference.